Mitochondria. Now let's think about the mitochondria. Unlike other organelles, mitochondria has a double layer of membranes. If you look at the structure of mitochondria more closely, mitochondria has an outer membrane and also it has an inner membrane. Both of them are phospho the bilayer the membranes. And that's why between two membranes there are intermembrane space. Also inside the inner membrane space we call matrix. Mitochondria's main function is digestion of glucose. The glucose molecule inside the cell is a breakdown into pyruvate structure, and this process is called glycolysis. Glucose is a 6-carbon sugar, and pyruvate is a 3-carbon other molecule. That's why pyruvate is almost a half molecule of glucose. After this pyruvate is formed, and then it can be converted into acetyl-CoA, and it enters into mitochondria, and then it enters into TCA cycle. Inside this TCA cycle, this acetyl-CoA is a breakdown and generate the energy materials NADH and FADH2. So from one glucose molecule, totally 6 NADH is formed and 2 FADH2 is formed. These high energy materials enter a series of reactions and eventually it generates high proton concentration in the area of intermembrane space. And this process is called as electron transport chain. And this high potential energy is utilized to make ATP. In the beginning, this kind of the fully utilizing the material to generate ATP, this ability is not available to every life forms. Only some bacteria developed this ability, and later this bacteria was adapted into our cells and it forms new organelles, so-called mitochondria. Now, if you look at more closely, we can find that inner membrane of mitochondria originated from bacterial membrane. Outer membrane, that's from our cell membrane. Wow. Also, mitochondria has its own genome inside. It's not only one copy, it has multiple copies. Not only genome, it has its own tRNA systems. It's totally different from our tRNAs. Other bacteria do not have this two-membrane system. Then if they want to generate ATP, they may pump out the protons to the outside of the cell. Then these cells cannot be captured in the defined space. They can be diffused out. In mitochondrial case, those are pumped out proton concentrations, they can be captured and maintained in the defined space. It doesn't need to be diffused out to the outside. So then definitely this can be much more efficient system than original bacterial system, we can think. Now let's think again about the life space issues. We can think cytosolic is a cell space, it's our space. And matrix is a bacterial space. Then in between, the intermembrane space is kind of DMZ, demilitarized zone. And that space has been used as temporarily keeping the high proton concentrations. And that is not the life space, that's the environmental space. Very interesting, isn't it?